Hi, in this video we're going to be looking at enzymes. Living things have thousands of different chemical reactions going on inside of them all of the time. Chemical reactions inside cells are known as metabolic reactions. The sum of metabolic reactions within a cell is known as the metabolism of that cell. Metabolic reactions need to be controlled. This is so that the reactions produce the correct quantity of the desired substances. Enzymes control the rates of reactions and they are biological catalysts. A catalyst is a chemical that speeds up the rate of a reaction without being used up in the reaction. As the catalyst is not used up in the reaction, it is available after the reaction to catalyze more reactions. We're going to be looking at how enzymes catalyze a reaction in more detail later on in this video. But first, let's have a look at cells and enzymes in more detail. A cell has hundreds of different chemical reactions that occur, and each reaction requires a different catalyst or a different enzyme. The nucleus of a cell contains genes, which control the rate of production of enzymes. These enzymes then catalyze certain reactions. Enzymes are made out of proteins. So this is how it works. The nucleus of a cell contains genes. The nucleus of the cell decides what enzymes to produce and in what quantities to produce them, and then when the enzymes have been produced, they then catalyze certain reactions. The activities of a cell are dependent on what enzymes it makes. So let's have a look at how an enzyme actually works. Most of the chemical reactions that occur in a cell involve the joining together or the splitting apart of chemicals. The molecule that an enzyme acts on is called a substrate. The enzyme has a unique shape that fits onto the substrate involved in the reaction. This section of the enzyme is known as the active site. So here we have an enzyme and a substrate. The substrate is the orange diamond and the enzyme is the blue shape. The enzyme is made out of proteins. The enzyme has an active site and the arrow indicates where this is. The active site will be a shape so that the substrate fits into it. The chemical reaction that's going to take place in this example is that the molecule that is the substrate is going to split into two smaller molecules, and the enzyme is going to catalyze this reaction. There are 4 stages to an enzyme catalyzing a reaction. The first stage is an enzyme being free to catalyze a reaction. The second stage is whereby the substrate fits into the active site of the enzyme and the substrate attaches itself to the enzyme. The third stage is the reaction taking place and the products are formed. In our example, the chemical reaction taking place is a substance splitting up into 2 smaller substances. However, it could be the case that the chemical reaction taking place is the joining of 2 substances together. When the substrate is attached to the active site, a lower amount of energy is needed to start off the chemical reaction, thus allowing the products to be produced more easily. The final stage is stage 4. After the products have been produced, the products are released and the catalyst is free to catalyze another reaction. The process then continues again. The substrate enters the active site of the enzyme, it attaches itself to the enzyme, the chemical reaction takes place, the products are allowed to be released, and then the enzyme is free to catalyze another reaction. But why are enzymes actually needed? Enzymes are needed in cells because the temperature in cells is too low for a sufficient amount of chemical reactions to take place to support life. Therefore, an enzyme is needed to increase the rate of reactions, thus allowing a sufficient quantity of reactions to take place to support life. The cells in a human body are at a temperature of 37 degrees, which produces too few reactions, hence why enzymes are needed. This brings us to the end of this video. For more content, quizzes and past papers, go on to www.elevise.co.uk.